Good day, everyone. Here's Group 4 presenting the aspects of gene therapy. But first, what is gene therapy? Gene therapy is an experimental technique that uses genes to treat or prevent disease. It is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effects of a disease-causing mutation. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. Meaning to say, patients don't need to take medications to cure their disease. It aims to treat the cause rather than the symptoms of the disease. Gene therapy is made to improve a patient's health by correction of mutant phenotype. A mutant phenotype is a gene that is rare and usually harmful. In contrast to a wild-type gene, it is not necessarily generated recently. This technique is widely used to treat those defective genes which contribute to disease development. Gene therapy is a novel treatment method which utilizes genes or short oligonucleotide sequences as therapeutic molecules instead of conventional drug compounds. Like what I've said earlier, the disease is treated with minimal toxicity by the expression of the inserted DNA by the cell machinery. It involves the introduction of one or more foreign genes into an organism to treat hereditary or acquired genetic defects. So, the prospect of human gene therapy was first realized in 1971 when the first recombinant DNA experiments were planned. Gene therapy can be simply viewed as inserting bits of foreign DNA into a patient's tissues in hopes of evoking a biologic response that will effectively eliminate the targeted disease. Major advances in recombinant DNA technology have occurred over the last 20 years so that now gene therapy is becoming a reality. Clinical gene transfer had its official beginning when, in 1989, 5 patients with terminal melanoma were given autologous lymphocytes that had been marked ex vivo, which would be discussed later, with a gene encoding resistance to the antibiotic G418. This study was designed primarily to trace the cells in the patient's bodies and to show the safety of gene transfer, and in that sense, it was successful. No helper viruses were found, no reverse transcriptase activity was detected, no toxicity was experienced, and the transduced cells remained otherwise normal. So the researchers are testing approaches to gene therapy, which are the in vivo gene therapy and the ex vivo gene therapy. The in vivo gene therapy is a direct method of inserting the gene material into the targeted tissues, and transduction takes place within the patient's own cells. This therapy is administered directly to the patient. The targeted cells remain in their body. In other words, it is a direct delivery of genes into the cells of a particular tissue in the body. In vivo gene transfer can be accomplished by 2 classes of vectors. A vector is any agent which carries and transmits an infectious pathogen into another living organism. These 2 classes of vectors are viral and the non-viral. The viral vector use replication in competent viruses such as adenovirus to deliver genetic material to target tissues. Because these vectors are based on viruses that have evolved specifically to deliver genetic material to mammalian cells, they usually result in a high percentage of cells being transfected. These vectors are so efficient that approximately 85% of all current gene therapy clinical trials use viral vectors. Unfortunately, they also suffer from limitations such as limited packaging capacity, immunogenicity, and an inability to transfect non-dividing cells. Non-viral use synthetic or natural compounds or physical forces to deliver a piece of DNA into a cell. The materials used are generally less toxic and immunogenic than the viral counterparts. Both these systems have distinct advantages and disadvantages, and the choice of vector used largely depends on the suggested application. In vivo gene therapy benefits from its simplicity and the reduced volume of material introduced into the brain, according to Freeman on 1997. These properties make it an ideal method when the target is a specific small area of the brain. This technique presents the following risk: the unwanted immune system reaction, because your body's immune system may see the newly introduced viruses and intruders and attack them. This may cause inflammation and, in severe cases, organ failure. It has also been suggested that replication-competent recombinant viruses may form due to random mutagenesis. The possibility exists that viruses which integrate themselves into the host genome, such as retroviruses, may cause accidental malignant transformation of the host genome. Inflammation and immune response are also of particular concern, even in an immune-privileged organ such as the brain, according to Hisht et al. On 2002. The in vivo method is the most straight tactic for gene transfer, and human in vivo studies for monogenic disorders have directed the vector containing the therapeutic DNA either directly to the organ of interest or into body vessels that nourish the organ. The in vivo can only be possible option in patients where individual cells cannot be cultured in vitro in sufficient numbers. And examples of in vivo gene therapy is in patients with cystic fibrosis. A protein called cystic fibrosis transmembrane regulator, or CFTR, is absent due to a gene defect. In the absence of CFTR, chloride ions concentrate within the cells and it draws water from surrounding. This leads to the accumulation of sticky mucus in respiratory tract and lungs. Treated by in vivo replacement of defective gene by adenovirus vector. Therapy for cystic fibrosis. Therapy of cystic fibrosis. Another is the ex vivo gene therapy. This is a technique where genetic manipulation of cells is undertaken remotely and more safely since it is outside the body. Therefore, unlike the in vivo gene therapy that is directly administered while the cell is still inside the body, the ex vivo therapy removes the cells from the body first to have safer manipulation, and it is being treated outside the body. An emerging therapeutic strategy particularly well suited to targeting a specific organ rather than for treating a whole organism. Transfer of genes to cultured cells and reinsertion, and it isolates cells with genetic defect from a patient. It grows the cells in culture and introduces the therapeutic genes, and then select genetically corrected cells and grow, and transplant the modified cells on the patient. Examples of ex vivo gene therapy is the first gene therapy to correct deficiency of enzyme adenosine deaminase, or ADA, that is performed on a 4-year-old girl, Ashanti De Silva, who was suffering from SCID severe combined immunodeficiency caused due to defect in gene coding for ADA. Deoxyadenosine accumulates and destroys T lymphocytes. The major advantage of ex vivo gene therapy is that the expression of the gene and the health of the transfected cells can be verified before it is introduced to the patient. This method can be useful for therapies where compounds should be released or expressed by specific cell types that can be selected on the culture dish before transplant. At this time, drawbacks include the sparse availability of suitable cells for transplantation, low survival rates of grafted cells, potential for migration and proliferation, in particular for stem cell lines, the possible need of immunosuppression, and the time-consuming and complex process of preparing the cells. An alternative are polymer encapsulated cells genetically modified to deliver the product of interest, which stops the possibility of migration, immunological response to the vector or engineered cell, or no immunosuppression is required, and provides a container that can be replaced or retrieved in case of adverse effects or low efficacy. Its limitations include the relative large volume size of the capsules, low cell survival, and restricted distribution of the gene product to the area surrounding the capsule. The ex vivo approach is restricted to disorders in which the significant cell population can be removed from the affected individual, modified genetically, and then replaced. Ex vivo strategies are best suited to applications in which the corrected cells can be easily obtained, for example, bone marrow, or when the corrected cells have a discerning benefit when they are back to the patients. There are also two types of gene therapy, which are the somatic cell gene therapy and the germline gene therapy. The somatic cell therapy or somatic cell gene therapy involves obtaining blood cells from a person with a genetic disease and then introducing a normal gene into the defective cells, according to Coutts on 1998. This type of gene therapy does not prevent the disease from occurring in the next generation because it does not affect the sperm and egg cells. Somatic cell gene therapy only affects the other body cells, meaning the affected person would be the patient only. If the disease is hereditary or could be inherited, then the offsprings might still suffer the same disease. Somatic cell gene therapy has to be done several times over the course of the patient's life because the effects do not last very long, such as cells of the bone marrow, and hence the new DNA does not enter the eggs or sperm. The genes transferred are usually normal alleles that could correct the mutant or disease alleles of the recipient. Somatic cell gene therapy involves the placement of a human gene into a living person's somatic cells, or cells that do not produce the eggs and sperm that in turn produce the next generation. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient's descendants. The technique of somatic gene therapy involves inserting a normal gene into the appropriate cells of an individual affected with a genetic disease, thereby permanently correcting the disorder. Somatic gene therapy is seen as a relatively unproblematic method from the ethical, not medico-technical point of view. since the transferred genes are not inherited by the descendant of the treated individual. So this figure outlines the simplest methods of getting genes into the person's cells using either viruses, which carry the human gene in place of one of their own genes into a cell, or liposomes or small fat-like molecules which can carry DNA into a cell. In some cells, the gene or genes become inserted into a chromosome in the nucleus. The target cells might be bone marrow cells, which are easily isolated and reimplanted. Bone marrow cells continue to divide for a person's whole life to produce blood cells, so this approach is useful only if the gene you want to deliver has a biological role in the blood. Next is the germline therapy. Germline gene therapy is when DNA is transferred into the cells that produce reproductive cells, eggs, or sperm in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. Because it targets the reproductive cells, any changes made to the DNA will be passed on to the next generation, unlike the somatic cell therapy that would only cure the patient itself. Consequently, the practice has dramatically divided opinion. Germline gene therapy is when DNA is transferred into the cells that produce reproductive cells, eggs, or sperm in the body. Germline therapy takes place in the reproductive cells. It involves genetic modification of germ cells that will pass a change into the next generation, meaning the future offsprings of the patient would be free from suffering the same disease. This type of gene therapy only has to be done one time to be permanent. One type of germline therapy is to treat a pre-embryo that carries a serious genetic defect before it is placed back in the mother by in vitro fertilization. Another germline therapy is to treat adult sperm and egg cells so the genetic defect is not passed into the children. In most countries, germline gene therapy, because of its potential effect on future generations, is appropriately outlawed. Our limited understanding of the complex interactions that have shaped human evolution, together with societal and cultural considerations, precludes the possibility of conceiving of responsible programs for germline genetic modifications in humans, as well as germline gene therapy approaches. Because people who would be affected by germline gene therapy are not yet born, they can choose whether to have the treatment because of these ethical concerns. The US government does not allow federal funds to be used to research on germline gene therapy in people. The idea of germline gene therapy is controversial. While it could spare future generations in a family from having a particular genetic disorder, it might affect the development of a fetus in unexpected ways or have long-term side effects that are not yet known. And using germline editing for reproduction is actually prohibited by law in more than 40 countries and by a binding international treaty of the Council of Europe. Next is the application or uses of gene therapy, which would be presented by Germline Oro.
And now let's move on to the application or uses of gene therapy. So gene therapy can deliver to target cells genes that code for the missing biological factor. Cancer, infectious diseases, cardiac disease, neurological disorders, and some inherited conditions are among the areas into which gene therapy research is being carried out. So gene therapy was made or discovered in order to cure such diseases, this kind of diseases. But as the base of gene therapy broadened, Other human disorders with genetic alterations have also been tried for treatment, like rheumatoid arthritis, various types of cancer, cardiovascular diseases, renal disorders, hepatic disorders, and even acquired immunodeficiency syndrome or AIDS. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. So, gene therapeutic techniques have recently been attempted to treat patients with the genetic diseases severe combined immune deficiency or SCID, cystic fibrosis, and Duchenne's muscular dystrophy. The optimist foresees a time when a patient will simply receive a snippet of DNA and go home cured. There are many ethical and scientific hurdles that must first be crossed for such a dream to become reality. So, this gene therapy is really a quite long process, Patricia. The technology has advanced so rapidly that many ethical questions weren't originally addressed and accordingly are now becoming the center of attention regarding human genetic research. So we have to know that there are a lot of ethical questions that will come up. So furthermore, scientists must find a way to outwit the body's immune system, which is primed to fight any foreign material such as inserted genes. There are also difficulties in getting the targeted cells to open up their molecular locks to allow the foreign genes inside. Gene therapy, like other medical advances before it, will have numerous failures before reaching its full potential. It will be important for the public, press, and medical industry to be patient in waiting for the dream of gene therapy to become a reality. So, as what I've said, gene therapy is really a long process. When a normal gene is inserted into the nucleus of a mutant cell, the gene most likely will integrate into a chromosomal site different from the defective allele. Although that may repair the mutation, a new mutation may result if the normal gene integrates into another functional gene. If the normal gene replaces the mutant allele, there is a chance that the transformed cells will proliferate and produce enough normal gene product for the entire body to be restored to the undeceased phenotype. Although gene therapy is a promising treatment option for a number of diseases, including inherited disorders, some types of cancer, and certain viral infections, the technique remains risky and is still under study to make sure that it will be safe and effective. Gene therapy is currently being tested only for diseases that have no other cures. So these are the uses of gene therapy: clinical gene transfer applications, vaccine development, production of transgenic animals, enhancing the resistance of plant, treatment of cancer and AIDS, gene discovery, gene therapy, of course, genetically modified organisms. So those are the uses of gene therapy. And now let's move on to stem cell therapy. What is stem cell therapy? Stem cell therapy, also known as regenerative medicine, promotes the repair response of diseased, dysfunctional, or injured tissue using stem cells or their derivatives. It is the next chapter in organ transplantation and uses stem cells instead of donor organs, which are limited in supply. So, stem cell therapy is really quite very important and useful. So now we are donating organs is very kanang famous. So this stem cell therapy will kanang instead of donating organs, they will use cells to transplant organ instead of donating organs. So the sources of stem cells: first, somatic. Somatic stem cells are the adult stem cells that occur inside the specialized tissues including bone marrow, skin, skeletal muscles, brain, liver, pancreas, and dental pulp. So these somatic stem cells are found in our body, specifically in bone marrow, skin, skeletal muscles, brain, liver, pancreas, dental pulp, etc. So these cells are multipotent and can only differentiate into the several types of functionally related cells that belong to the tissue of origin of the stem cells. Therefore, they continuously divide to produce new cells. A part of these new cells differentiates into the functionally specialized cells in the tissue, and the rest of the cells renew the existing stem cell population, as what you can see in the kanang image or the illustration picture beside. It is the locations of somatic stem cells in the body. So their brain, the teeth, blood vessels, skin, heart, liver, bone marrow, etc. Somatic stem cells occur in most of the organs of the body, including bone marrow, skin, skeletal muscles, liver, etc. So that's what I've said earlier. Somatic stem cells are present in many fetal and postnatal tissues. Somatic stem cells are also capable of self-renewal and, with appropriate signals, differentiate into various cell types from the organ from which they are derived. The extent to which they are capable of differentiating into cell types from alternative lineages is controversial. So next is embryonic. These are the cells in the early stages of the embryo. The zygote, which is the conceptus of fertilization, divides by mitosis, forming the morula. After 5 to 6 days of fertilization, the morula develops into the blastocyst that contains two parts, the trophoblast and the inner cell mass. The trophoblast is the outer layer of the embryo, which contains cells that develop into the placenta and umbilical cord. Here, the cells in the inner cell mass are pluripotent and are capable of differentiating into any type of cells in the body. The embryonic stem cells in culture are capable of self-renewal without differentiation and are able to differentiate into all cell types of the endoderm, mesoderm, and ectoderm lineages using appropriate signals. In utero, the blastocyst implants and all three embryonic germ layers are formed during gastrulation. Embryonic stem cells are derived from the embryo and have the potential to become all the different cell types of the body, or what we call pluripotency. Now let's move on to the application or uses of stem cells. Stem cell transplantation has been established as a useful therapy for hematologic malignancies and some solid tumors. Some patients, however, still die in allogenic SCT because T lymphocytes cause fatal GVHD, and many are not cured in autologous SCT because disease recurs post SCT. Disease may occur because the tumor was not completely eliminated or because the stem cell component was contaminated with tumor cells that home to sites of prior disease. Stem cells have been applied in the treatment of serious diseases for more than 55 years. They are applied especially to treat cancers, which require high dose chemotherapy within the scope of medical care. The patient's own stem cells are extracted from bone marrow or peripheral blood prior to high dose chemotherapy, stored temporarily, and transplanted after the treatment in order to minimize the side effects of the aggressive chemotherapy and to support the regeneration of destroyed cells. Besides cancer, several hundred thousand people come down with common diseases like dementia, which belongs to the neurodegenerative diseases, cardiac infarction, stroke, arthritis, or diabetes every year. The lifelong therapy causes enormous cost in the healthcare system. Stem cell therapy offers great potential for the treatment of such diseases. Experts expect that every seventh person up to the age of 70 will need a therapy based on stem cells in the future to regenerate sick or aged cells and tissues. Stem cells have already been applied successfully for hematopoietic disorders, acute and chronic leukemia, myelodysplastic syndrome, lymphomas, aplastic, sickle cell anemia, beta thalassemia, immunodeficiency, SCID, Wiskott-Aldrich syndrome, metabolic disorders, mucopolysaccharidosis, cancer, and multiple myeloma and neuroblastoma. So these are the applications of stem cells.
Good day, everyone. I was assigned to Bioethics of Gene Therapy and Status of Gene Therapy in the Philippines. Gene therapy in human beings raises a variety of important ethical, legal, and social issues. The success of gene therapy led to boom into the development of several studies with prospects for therapeutic use of genes, and soon after April 14, 2003, the day that the mapping of the human genome was completed, it became possible to diagnose and treat many diseases of genetic origin before they process using gene therapy. However, these findings have triggered a series of bioethical controversies about the genetic safety of genetic engineering and the likelihood of its use for purposes of eugenics. Gene therapy is a therapeutic strategy characterized by the transfer of genetic materials in order to rearrange the genome of the target cells to enable expression of the inserted gene or inhibit expression of a specific gene. Among the methodologies used in gene therapy, there is application of RNAi widely used in biomedical research in recent years due to its relative ease of handling. Quality of life is a concept that has been extensively studied in recent years and integrates different fields of knowledge, especially those included in health sciences such as medicine, psychology, and biology, and more. In this context, medicine presents itself as primarily mediator in order to prevent diseases and to maintain good health of the patient. Science cannot afford to use still unfinished therapy such as gene therapy at this time, since it is still experimental. With the advances made in the biotechnology in recent decades, the clinical responsibility of medical practitioners and the debates about intervention methods used by them has been inevitably increased. The participation of everyone in order to contribute their views on the bioethical issues inherent in aiming analyzing the risk of expected benefits of gene therapy is indispensable. So today, to be part of that context and help to guide better decisions, rules, deontological ethics are not enough, and there is the need for plural interdisciplinary reflections, always focused on human dignity. Although the advantages of sequencing the human genome have been widely accepted, there are still ethical concerns regarding those benefits, and several problems were encountered in clinical protocols, making it clear that we still have a long way to go before employing technology, RNAi, in modern medicine. From the perspective in bioethics, the use of gene therapy appears to be closely related to several negative factors, among which can be cited economic difficulties, particularly with regard to health distribution, political and cultural conflicts, as well as scarcity of the studies evaluating the impacts of the use of gene therapy on human health, clinical dilemmas, and legal issues. Some methods, including RNAi, are associated with the use of pathogenic, however attenuated, virus as transferred vectors. The initial proposal for the use of viruses as carriers to transport and introduce genes into the patient seems to be very simple. However, some of them are associated with severe human disease, such as viruses causing pharyngitis, conjunctivitis, leukemia, and AIDS. The use of viruses belonging to the lentivirus family, which includes HIV, seems very dangerous, because with the exception of American communities, the global population does not have antibodies against this pathogen. Overall, gene therapies are new procedures that are still in the experimental stage. This is a very risky therapy since many vehicles are viruses with genetic materials consisting of RNA, and therefore they can more readily undergo genetic recombination and become more virulent. From the point of view bioethics, the main obstacle to the application of RNA-based gene therapy is the fact that the safer non-vectors currently available are still inefficient or have very limited application. Therefore, studies that evaluate the effects in experimental models and in preclinical trials are needed in order to validate the potential effectiveness of this type of therapeutic intervention. It is necessary to evaluate the actual benefits as well as to detect the potential risks of implementing this therapy so that both safety and human health are preserved, and worse health problems than those we have today are avoided. The speed and effectiveness of some of the protocols used in gene therapy have not allowed much scientific and legal discussions about experimental regulation and commercialization or about the long-term effects of this new therapeutic approach. This decade is essential to further understanding about the side effects arising from the clinical trials in order to intervene if necessary. New experimental designs to determine the faults and dangers arising from the use of gene therapy before their deliberation, and hence the application as routine technique in the hospitals and health centers are therefore timely and indispensable. In 2008, the number of clinical trials using gene therapy as an intervention method reached 118, and since then, there has been a decrease in these experiments, probably be due to temporary restrictions, omitted flows in the protocols, or even bioethical issues. Over the past 4 years, clinical trials using gene as therapeutic mechanism were recorded in more than 33 countries with representatives from all continents, although the American continent is responsible for over 50% of the trials currently in progress. It is worth saying that the numbers may be underestimated due to the secrecy of information made available and omission of data in some countries. The vast majority of clinical trials using gene therapy is currently connected to cancers, 64.3% with a total of 1,223 trials, monogenic hereditary disease, 8.8% with 1,667 trials, heart disease, 8.3%, 158 trials, infectious disease, 8% with 153 trials, neurological disease, 1.9% with 36 trials, optical diseases, 1.5% with 28 trials, inflammatory diseases, 0.7% with 13 trials, and other negative factors associated with genetic disorders, 6.5%, 119 trials. In global legal context, the use of experimental trials with the inclusion of intrinsic parts of animals, plants, and other organisms, including DNA, RNA, and stem cells in humans, was always challenged to face dilemmas in security, especially about the consequences of the development of biomedical sciences and biotechnology. The idea is defended in both international sphere and constitution of many countries, in addition to the statutory law including the subject. In this context, it is vital above all to understand the principles of bioethics and ethical science align with their socio-ecological responsibility. So, to prioritize the health and welfare of man and nature in order to properly utilize the natural resources and technology. Among the fundamental principles, bioethics and maintenance life and vital processes with an ongoing commitment to transparency and the dissemination of knowledge involving biological and medical sciences. The law relating to use gene therapy is rarely debated in scientific circles because of the great resistance on the use of these clinical trials despite of numerous prospects for curing different diseases. However, such perspective of submerged in dubious methods and effects of the projects now under study are not predictable. On the other hand, men are entitled to life and health are stated of the Universal Declaration of Human Rights. On the other hand, there is There's a question to what extent we can risk on behalf on still unfinished scientific research. As in other areas of research, validation of the new therapeutic methods is closely related to development of clinical trials and prior approval by local, national, and international ethics committee is therefore required. Some types of vectors, notably adenoviral and retroviral vectors, have produced serious and even fatal side effects. Therefore, security seems to be the main obstacle of the application of this type of clinical intervention in hospitals and other public health care centers, particularly in the underdeveloped areas or countries. Not so far from the new technological possibilities applied to modern medicine, many matters involving moral and ethics were raised with heated debate, especially on the behavior of the professionals involved, including doctors, researchers, patients, and other people involved with the problems of medicine and public health. Status of gene therapy in the Philippines. Among the genetic tests, NBS is the only one covered by the National Health Insurance. All other expenses related to treatment upon diagnosis are out-of-pocket expenses for the family. The following are the barriers to accessing genetic service in the Philippines. First, financial, as most family cannot afford out-of-pocket expenses for the expensive genetic testing and treatment. Second, geographical, being an archipelago of 7,107 islands. Third, lack of awareness among different stakeholders, that is, health professionals and parents. Fourth, compromise access to genetic service at the regional and provincial level. And lastly, lack of geneticists and genetic counselors. The integration of NBS is public health system has been opportunity for introduction of genetic services in the 17 regions of the country. Currently, there are regional DOH NBS coordinators assisted by a full-time NBS nurse and coordinators who are also involved in other genetics related activities such as birth defect surveillance and the telegenetics referral system, which aims to further expand the delivery of genetic services in the country. Human resources and training in medical genetics of nationally there are only 10 trained geneticists. 8 are in Metro Manila and 2 are in the provinces, 1 in Davao City and 1 in the Cebu City, making a medical geneticist to population density ratio about 1 to 11, 751,625. To date, all clinical geneticists are pediatricians. Medical genetics has been recognized especially since 2000. There is no separate department of medical genetics. Only the Department of Pediatrics at the PGH has separate section for clinical genetics in the whole country. The PGH Department of Pediatrics offers 2-year fellowship program clinical genetics designed to provide broad clinical exposure to areas of dysmorphology, biochemical genetics, cytogenetics, molecular genetics, and neonatal screening programs. Since 2011, the fellowship program requires its fellows to pursue an MS in genetic counseling. The University of the Philippines, Manila started the 2-year Master of Science in Genetic Counseling program in 2011. As a response to the growing need for counseling services, the 2 major DOH programs, the SBS program and the BDS program, it opens to nurses, doctors, and other allied health professionals. That's all. Thank you.